John, and then we can get started.
Hello, good evening, sir. Hello.
गुड इवनिंग
हाँ सर, अभी वक्त है थोड़ा प्रोग्राम स्टार्ट करने में सर.
So we'll start within next 2 minutes.
Okay then, definitely sir, we will wait. Okay, it doesn't matter. Okay.
Fine.
Yes.
Assalamualaikum, sir. A very warm good morning to Dr. Tanveer. Can you hear us, ma'am?
I am trying to adjust my headphones. Can you guys hear me?
We can hear you, yes.
Oh, okay.
AIC
कितनी देर लगा दिया इसका? बहुत लंबी लाइन थी. कहाँ लाइन थी? बाथरूम में.
Let me check my bandwidth. I may be able to come on video.
Sure.
But if it is maybe after the introductions, I will go off of video to save
That's
bandwidth.
fine. Yeah, that'll be fine, ma'am. That'll be fine. We'll get started now. I think Professor Abu Sufyan, we can get started. I think Director Sir has joined in. We can get started, sir.
Okay, can we start, sir?
Yes.
Okay. Assalamu alaikum and a very warm good evening to everyone. I hope you are safe, healthy and doing good at your end. We all are looking forward to come out of this difficult time of COVID-19 pandemic and looking for something new and best to sharpen our skill.
Professor Sufyan?
We cannot hear you.
Yeah. I think his video goes freezed up. Professor Abu Sufyan, can you hear us? We got disconnected. Okay. Just wait for a couple of seconds, and if not, then I'll take over as host.
I think his net is not stable, I think.
Maybe, yeah. Okay.
Hello.
You can dial in.
Yes,
you can
yes.
ask him to just dial in.
Yeah, we got him back. Yes, Professor Sufyan, go ahead.
There was a connection issue.
Okay.
Okay, sir. Thank you. So to emerge as a successful teacher with recent updated research knowledge, multidisciplinary diverse skill and to serve expectation of all the students at the same time, ensuring own professional development as a researcher and innovator is a challenging. So this 1 week AICTE-IST sponsored faculty induction program on multidisciplinary and translational drug development with sub-theme of AI in healthcare is planned to unlock this trait of the teachers. On this note, I am Abu Sufyan, Assistant Professor at School of Pharmacy and Lead Coordinator of this program. On behalf of Anjuman Technical Campus, New Panvel, extend my warm welcome to all the dignitaries, resource person, faculty members from different colleges across the area to the day and inaugural session. To start with, as a tradition of Anjuman Islam, I recite and translate few verses from Holy Quran. Bismillahirrahmanirrahim. وَلْتَكُمْ مِنْكُمْ أُمَّةٌ يَدْعُونَ إِلَى الْخَيْرِ وَيَأْمُرُونَ بِالْمَعْرُوفِ وَيَنْهَوْنَ عَنِ الْمُنْكَرِ وَأُولَٰئِكَ هُمُ الْمُفْلِحُونَ. The translation of these verses from Holy Quran is, and let there always be among you a body of men who should invite to goodness and enjoin virtue and forbid evil, and it is they who shall prosper. These verses was from Surah Al-Imran of Holy Quran. Indeed, it is worth implementing in our life. Going forward, we have with us convener of this 1 week faculty induction program and in charge dean of School of Pharmacy, Dr. Sharik Sayyed. He has over 13 years of experience in industry and academia. He has completed doctorate from University of Maryland, USA. He worked at senior leadership role in BMS as a drug development lead for oncology medicine. He is the member of several prestigious organizations. He is also recipient of Pharma Ratan and Anjuman Islam Best Teacher Award. With this short introduction, now may I request Dr. Sharik to give opening remark and take further proceeding of this program. Dr. Sharik, it is over to you.
Yes, can you hear me properly?
Yes.
Okay, great.
You're audible.
Thank you, Professor Abu Sufyan. So once again, on behalf of Anjuman Islam's Khalsa Technical Campus, I would really like to warm welcome all my faculty colleagues, both in pharmacy and engineering domain, to this week-long faculty development program, which has been sponsored by AICTE and IST. We're very fortunate to have all of you coming from across the country. And I know all of us are going through a very challenging pandemic times, and I take this moment to pray for the health of all of you, your families, and the entire humanity. So a couple of months back, when IST asked us to host an FDP after December F1, we really wanted to explore an interdisciplinary domain. And when we talked to our International Advisory Board, we thought of narrowing down to the role of artificial intelligence into healthcare. And as you all would agree, artificial intelligence is rapidly affecting our lives, and sometimes in a very obvious manner, such like giving suggestions on YouTube channels to making big decisions on finances and like something like predicting COVID pandemic. So at School of Pharmacy, we've been very interested into exploring how artificial intelligence is affecting healthcare. And you probably would know that there's a huge amount of work that is going on in the entire ecosystem of healthcare, right from discovering new drugs to assisting physicians to patient management. And our goal with this FDP has been basically to invite experts who have been working in these domains to give a glimpse of the work. And ultimately, the idea is to spark a research conversation. So at the very outset, I'd really like to thank all our experts who have agreed to come to this whole week of faculty development program and share their work. I know they've been very busy, but they've still graciously agreed to come to this program. I know we've all are going through what is I call as the FDP and the webinar fatigue. But what we've been trying very hard to put together a program which is very valuable and gives benefit for the time that you have essentially with us in this FDP. So we've tried very hard, and we've been very lucky to have very good speakers who have come in and who will be speaking to us the week. So hopefully, this entire week of an hour and a half every day would be a time worth spent. To start formally, I would like to invite our campus director, Dr. Abdul Razak Honutagi, for his opening remarks. So before he comes on stage to speak, give me 1 second. Give me 1 second. Is my screen visible?
Yes, sir.
Yes, so Dr. Abdul Razak Honutagi is the founding director of Anjuman Islam's Kalsekar Technical Campus. He holds a bachelor's into civil engineering and a master's in civil engineering from IIT Roorkee and a PhD from IIT Mumbai. He's been a former principal of Amit Sabu Siddiqui Polytechnic, Mumbai. He has an extensive experiences of 32 years in the field of engineering, education, research, and innovation. He's got several outstanding research awards and recognitions. Some of them are the Jubilee Memento Awards and the Best Teacher Awards at Anjuman Islam, citations from Lion Club International, Lokmat Panvel, Gaurav Puraskar. So some of them just to mention. And I personally have a good fortune of working with him for over 5.5 years. And I found him a man of vision, a man of innovation. He's constantly looking for newer opportunities for innovation, and he's very passionate about bringing newer pedagogies into teaching and research. So it's my great pleasure to invite our very own campus director, Dr. Abdul Razak Honutagi, for his welcoming remarks. So over to you, sir.
Sir, you need to unmute yourself. Director, sir, you need to unmute.
Yes.
Yes, we can hear
thank you, Dr. Sharik, Professor Abu Sufyan. Indeed, a great pleasure to be a part of this FDP once again, and that too on a starting day where a very renowned and versatile person being invited as a keynote speaker, Dr. Tanveer Sayyeda Mahmood. We are indeed lucky, to have that kind of speaker, along with Dr. Richa Sharma from Mumbai, who is associate professor in University of Mumbai. So both these two persons on the day will definitely make us, learn. Especially the participants will be more delighted once they undergo in their presentation. I'm not here, as Dr. Sharik said, I am from the field of civil engineering, and being a campus director and being a part of this, FDP, I'm here to just introduce to our participants about our campus, our parent body. That is what my purpose is. But I don't know, how many participants attended first part of this FDP, and if there are same persons, and it will be a repeat for them. Otherwise, for new participants, it is like my introduction will be worth mentioning. We are managed and governed by a very renowned 148-year-old organization named Anjuman Islam, which was established in 1874 And this institution is standing with its own achievements and accolades, having 97 institutions, 1,10,000 students, and more than 6.5. This kind of institution, which has been a symbol of national integration and secularism. Now, under that worthy parent body, we have established very recently in 2011 this campus named Kalsekar Technical Campus, having multidisciplinary institutions like School of Pharmacy, School of Architecture, School of Engineering, and also a polytechnic, where around 4,500 students are undergoing their different types of studies, ranging from graduation, their undergraduate program, to PhD program. So, as such, in a short span of about 9 years, this campus has become a role model campus for Anjuman Islam, and there are a lot of achievements that all goes without saying, because of the dedicated, committed efforts of the entire fraternity of our faculty and staff. Name a few. As such, this particular AICTE IST funded program, which has come uh, uh, due to the effort which was put by Dr. Sharik, Professor Abu Sufyan and his team. So ultimately, the authorities sanctioned this project, and that is basically to make the fraternity of pharmacy to get the next level of knowledge and research avenues which are happening. So the idea of you all know, the idea is basically to develop the faculty members. So that development happens only when the relevant speakers are invited, and in a planned way, those speakers will make definitely some sense and also motivate the participants to undertake relevant research in the field of especially the pharmaceutical field. And accordingly, the topic for this FDP is very relevant. So we are into the age of AI, machine learning, biotechnology. So in this particular scenario of even the pandemic scenario because of COVID-19, all this makes sense. So therefore, AI-based training for faculty is more relevant, and therefore the speakers will make definitely a lot of meaning. I welcome all participants who are attending for this FDP, and also today's keynote speaker, Dr. Tanvira Syed Mahmood, and Dr. Richa Sharma, Dr. Sharik Syed, Professor Abu Sufyan, and all his team of School of Pharmacy to this FDP. And I must mention once again that Dr. Sharik, who has all that background experience of working in US, and that experience of industry is helping us a lot to add on or taking rather academic
So you need to unmute yourself. I think you got muted. Yes. Thank you. Yes, Professor. Yes, Abu Sufyan. Okay, so thank you, Razak sir, for the kind introduction for the entire campus, and it is a good fortune of us to be working at Anjuman Islam's Khalsa Technical Campus, which has roots with Anjuman Islam's and 140 years old organization with a very strong history into education as such. So without further ado, I'd like to welcome our first keynote speaker for the day, Dr. Tanveer Sayyeda Mahmood. She's an IBM Fellow and a Chief Scientist, overall lead for Medical Sea Radiology Grand Challenge project in IBM Research Almaden, and Medical Sea with an exploratory research project with global participation from IBM Research Lab across the world. And the goal of this project is to pretty much develop our automated radiology and cardiology assistants, and that could help the future clinicians in making decisions. Dr. Mahmood is graduated from MIT's AI lab in 1993 with a PhD in computer science, and prior to IBM, she has worked with research staff member at Xerox Webster Research Institute. And prior coming to IBM, and she's led an image indexing program at the Xerox Research and one of the first early originators in the field of content-based image and video retrieval. She's working currently on applications of content-based retrieval into healthcare and medical imaging. For the past 30 years, her research interests have been in the variety of areas related to artificial intelligence, including computer vision, image, video databases, medical image analysis, bioinformatics, signal processing. She has over 200 referred publications and over 80 patents filed. Dr. Mahmood will be the general chair of MICCA 2023, which is a premier conference in medical imaging. And she was a general chair in the first IEEE International Conference on Healthcare Informatics Imaging Systems in Biology at San Jose in California, 2011. She's also the program co-chair for CBPR 2008. Dr. Mahmood is a fellow of IEEE, and she's also a distinguished fellow at the Center for Artificial Intelligence in Medicine and Imaging at Stanford. She's also the first IBMer to become an AIBME fellow, a member of IBM Academy. She was declared a master inventor in 2011 and in 2019. She's recipient of key awards, including the IBM Corporate Award 2015, Best of IBM Award, and several Outstanding Innovation Award. So without further ado, I'd like to welcome Dr. Tanveer Saida Mahmood here. A very warm good morning to you, ma'am. I know it's very 6:45, 7 o'clock a.m. into Pacific Coast. Thank you so much for joining in an early morning here. And we all are delighted to have you here, and we'd like to hear from you.
Thank you very much for that introduction. I'm honored, and it's a great pleasure to be here to talk to your faculty. Let's see if I can, I would like to go off of video to preserve bandwidth, and hopefully the clarity of the presentation will still come through.
Surely, surely.
And then let me go share my screen.
You can see your screen now.
You can see it. Okay, great.
Great.
All right. So I was not aware of what title you guys had given. Sorry.
Can all participants mute all? Okay, I'll mute all of them, and ma'am, you can unmute yourself.
People can still hear me, right? So,
yeah, I can hear you, yes.
Okay, thank you. So I was not sure of what the title you guys have given, so I have tried to make it as close as possible to this, because the talk I'm going to give today is primarily for the radiology community. But hopefully it will give you an indication of what does it take to build an AI software that is a clinical workflow ready.
Sorry about that.
Okay, I have to keep doing this a few times. Okay, got it.
Okay. Sorry about that.
All right, so I am from the IBM's research lab in San Jose, California. You can see a beautiful campus over there. And IBM Research is the academic wing of IBM. And our goals are similar to faculty in terms of trying to advance the state of the field, and publications and presentations are an important part of our work. Okay. And I don't know who the audience members are who are turning off on this video, but could I please request that they go on mute? Thank you. So before I start, I want to acknowledge my team. There's a large number of researchers who have been working with us to pull off this grand challenge. We just concluded the challenge towards the end of last year when we published our work in JAMA. But one thing I wanted to point out over here is to pull off a project like this, you do need cooperation from clinicians. So a number of people on our team are radiologists, both in staff and in a crowdsourced form, along with medical imaging researchers, artificial intelligence researchers, and software engineers and statisticians and so on. So you can see a variety of talent that is needed to pull off a project like this. Okay, so let's see what is the grand challenge when we talk about. And so grand challenges are projects that IBM Research takes on every several years. This has been running for 5-6 years in the past. And some of them you have heard about, such as the Watson Jeopardy Challenge, and then the recent, the IBM Debater Challenge, in which the machines tries to debate humans. So this particular one was in the field of radiology and looking at the lives of radiologists who look at enormous number of images per day. It says my connection is unstable, so hopefully you guys can still hear me. The current diagnosis that they are doing is primarily based on imaging alone, and although they could potentially have clinical context of the patient from their EMR, this is not something that is easily done, and most of the time they are sitting in their dark rooms and making their decisions primarily based on a very small background information given, and the rest is up to whatever the imaging can reveal. So the idea of this challenge was to build a virtual radiology assistant similar to a teaching the resident in a teaching hospital, who would go ahead of the teaching clinician, collect all the necessary information about the patient, and essentially make a one-page summary so that when the clinician walks in, they can rattle off what they have learned about the patient in a very summarized form. And from there, it is easy for clinicians to make their diagnosis. So our goal was to produce such a radiologist assistant that could actually be trusted in the clinical workflows as we go. So to do that, we had to prove ourselves that we are good enough for use, and that's what the challenge was about, was to take on a goal that would be so high and difficult to achieve that this would take several years to complete, but also a concerted effort in which we see the role of a priori domain knowledge as well as learning from data using the latest AI techniques and bringing them all together in a reasoning framework. And so we saw this as a great application of all the different fields of AI for a practical use case. Now, the goal originally was to take the American Board of Radiology certifying exams, but as it happens in these grand challenges, these are large-scale, high-risk initiatives that potentially can lead to billion-dollar businesses for IBM. So it's somewhere along the line, as we were doing our project, we got an opportunity to acquire a company called Merge Healthcare that was already in the PACS business. And so this project's successes came on earlier than we were expecting, right in the middle of our conducting our project, and when this company was bought, and that became Watson Health Imaging, which has now launched some flagship products that come from the research that was done in this project. However, I'm not here to talk about those products or this being an academic forum. And so what we will do is we continued on with our challenge, but this time focused on a use case that the business also indicated was a good one to focus on and just as difficult, and which was to basically take a Turing test for radiology by the machine and do it on a topic that was focused enough and complete enough. So the idea was to do a full preliminary read from a modality, which is going from an image to a report. So to give you an example, you have a chest X-ray, and you want to be able to produce an automated report that documents all the different findings that are seen in chest X-rays and in an English language rendering as well. And so the goal would be to detect all possible findings and flag as few incorrect findings as possible. Now you might say, why chest X-rays? Now, we chose this modality for a number of reasons. First of all, this is the most prevalent imaging exam, the most common one done in emergency rooms or in inpatient settings. And so there is a huge need for looking at this from a prevalence standpoint. These are also cases where there is a need for an improvement in interpretation because of the cognitive system and biases and system factors that occur that leads to many overcalls or missed findings. For example, knowing from here which of these is actually a mass or a nodule is difficult to interpret, and there's as much as 26% error rate quoted for radiologists. So these are hard enough even for interpretation by radiologists. So we thought this would be a challenge for machine as well. The idea being still of assistance, so that if the machine can detect all these findings, and clinicians still have the ability to force signing off and make an alert. So the way we envisage how this will change the future is currently the practice is very dictation driven, at least in the United States. The radiologists dictate their reports. Either this dictation is outsourced for transcription, but more commonly it is now read by speech recognition software and transcribed, and then the clinician will still have to read the report, corrected prescription. Nearly 85% of the time, this is something that is done. So now the automated reads of the future we would have the imaging come in, and technician, in fact, can itself benefit at the point of itself, and whether when imaging, once it is passed, you have to be read first or routed to the relevant. This templated read is already available, and it's something that they can use to fill out a report as we go. So this can be used at any of the steps in a clinical workflow today, and having a service that you can send out for preliminary reads would we feel will be a very disruptive technology coming in future for that is heavily dominated by dictation-driven workflows currently. So to give you a flavor for what is possible, let me run the service itself. So here you can see an image, and then when you run it through our service, you would be able to do the analysis of the various structures, and then produce a report that says this one passes technical assessment. And you can, many of the findings are usually rule out findings, so there are negative findings in there. And this can be done for any chest x-ray that is in your packs, or something that you have taken from your device and relayed to this service. And you can see this is a poor quality chest x-ray from a portable machine. And you can see the, in this case being an inpatient chest x-ray, there is tubing present as well. And the system is able to report on that. So this gives you sort of a flavor for what is possible. Now, as we said, this was supposed to be a grand challenge. So let's look at some of the factors that make it a grand challenge. One of them is the number of findings. So when we say it is a preliminary read and it is to be practically of use in a clinical workflow, we can't just do one or two findings, which by the way is the most common practice in AI and radiology today, to focus on one or two findings, partly due to the regulatory framework we have from FDA in getting allowance for pre-market clearance. But the reality is, if you want it to be useful in a clinical workflow, you can't just have one or two things done by machine, and then the rest of the things it's still left to the radiologist. So they don't see as much of a benefit. So doing a full read meant covering as many findings there are in a chest x-ray. But who has cataloged how many findings there are? And that is the first problem we encountered, is if you want to take on a challenge, there's a lot of work to be done on the academic side first in terms of this type of cataloging. The second was to make sure that they are as accurate as possible in detecting the findings. So then we needed a benchmark. We needed someone to tell us how good are the radiologists. It turns out that there are no good studies for covering a wide spectrum of findings to say how good the radiologists are. Now, we mentioned numbers such as 26% error rate, but those were for specific conditions. So if you wanted to attempt a quantitative comparison, you needed to first set up a benchmark. And this by itself is a challenge because this requires radiologist cooperation to even record these accuracies. Now, once we get that, we need to get to describing them in a report, which means the reports have to be fairly fine-grained in quality. So you can see some examples of reports, sentences that are found in radiology reports, such as there are calcified right hilars and mediastinal lymph nodes, which points to the location, the laterality, the severity, and so on. So the machine should not just be able to find a label such as pneumonia or opacity, which is currently what is being done today, but to be able to describe that finding at a fairly granular depth, indicating all of these details as well in order to be valid as a report. And of course, the language has to come out right as such. So many of these factors make this a grand challenge. So as you can see from here that we had to basically address problems both on the radiologist side and setting up the benchmark, cataloging the findings, generating the knowledge itself, as well as in developing a machine that would meet the benchmark or exceed the benchmark in terms of understanding the findings as well. So there's a variety of data science problems that need to be solved in developing these type of projects, and I've itemized a few of them, and we will cover these as we go through as such, and you can get a flavor for what does it mean to build an AI solution in healthcare. So let's start first with the cataloging the findings itself. The key teaching points over here is that it's very important to characterize the full spectrum of findings, and developing such characterization requires a large-scale effort, a concerted effort, primarily by clinicians, but the process that they can use can also be changed from the traditional textbook-driven process to a more data-driven process as well. So this is something that we did in our methodology. So let's first look at, starting from the basics, how does a radiologist interpret the findings and what are the sequence of steps that they're going through? The first thing is to make sure that the image is ready for interpretation, which is a technical assessment problem, knowing whether this is qualified for interpretation itself. So is it of diagnostic quality or non-diagnostic? And if it is not diagnostic, what are the problems seen? And these have to be documented in the report as well, so that a repeat imaging, if necessary, can be taken. The second is to figure out what is the viewpoint and position. Is it an AP, anterior, posterior, anterior views, or a portable? And this requires, and this also sets the a priori for them. If it is an AP view, it's probably inpatient, which means the patient is already in here with a problem. And in that case, maybe it is the importance of the tubes and lines that are placed in him that need to be detected and described to see if they are properly positioned, or if there are any artifacts or devices such as pacemakers or sternotomy wires that need to be described as well from positioning and status standpoint. And this could also be an anatomical abnormality, which in this case opacities, and then or such abnormalities that may lead to a disease. Although the differential diagnosis is done a little bit later, the radiologist does also give indications from a differential standpoint for abnormalities, and the overall information goes into a report. Understanding this radiologist workflow is important for cataloging the findings, but also giving a methodology for building machines that follow the radiologist process. So the next step was to catalog all the findings, knowing that these are the different types of findings. And yes, we took a combined approach, first starting from knowledge where the radiologist provided, and but then curating a large set of reports derived from about 200,000 radiology reports. We cataloged the different types of findings using a semi-automatic interactive learning tool that I will briefly talk about. But you can see from here that the result of it is a very large catalog of findings, and the dictionary itself that we developed has over 11,000 terms that describe the many possible ways in which these findings are actually cataloged in reports. So the tool itself is a machine learning tool that uses a semi-supervised learning process where text analytics and embedding schemes are used to find terms, starting terms, which the clinician then guides as being positive or negative through a reinforcement learning type approach. And then the system makes further predictions, and the predictions of the system get more and more accurate with this type of supervision. And with this, we were able to quickly assemble over 11,000 terms representing different types of findings as we go. Now, here is a catalog of the core findings that we were able to locate. And this, as you can see, is probably the largest recorded from a core finding level for the type of findings seen in chest X-rays. And those that are clinicians in the audience or radiologists can relate to many of these findings, but just having them cataloged and described at the next granular level was an achievement from a clinical knowledge and contribution to the medical community standpoint. Okay, so the next step was to prepare the machine for recognition of these findings. And in doing so, in order to cater to different populations, it's important to acquire data sets from multi-institutions. And to essentially create an environment so that you can develop these data sets and make sure that the training and testing and validations you're doing are on sufficiently different prevalence distributions to give you the kind of robustness that is needed as such. Now, of course, there is the huge problem of labeling these data sets, so you could use the supervision approach, because even though there is unsupervised learning, when you're starting with such large collection of findings, you need at least a supervised approach to get you going first and after those models are built, you can perhaps experiment with some pre-trained models, biased approaches. So for us, we collected the data sets from about 3 different sources, mostly from US sources, but across different demographics zones from the East Coast and Northeast and so on. And the number of patients and the kind of prevalence distributions you can see over here. Now, in order to test the algorithms and for benchmarking purposes, we separated them from a different data set. And to test the robustness of this for field data sets, which are representing deployment situations, we actually conducted this in international settings at the Deccan Hospital in Hyderabad in India, where they supplied around 10,000 images. We conducted both retrospective and prospective studies at this site to further improve and catalog our performance. And then the last study, which is the Turing study that was done to show that the machine has reached this capability, was also done with a different curation process, ground truth data set that I will talk about. Okay, so once we have the data sets, now we need to label them, which means we need to take the large number of findings that we have cataloged and see which of them are actually present in these images. Now, this could be a huge effort when we're talking about millions of images. So the first approach we tried was to have clinicians label these images by cataloging all the findings and asking them to select among these findings, or to basically describe them in dictated reports from which we could try to analyze the findings. Now, as it happens, this process, we ran this as a crowdsourced platform with over 40 radiologists around the country participating. And in about 4 months, we could label about 36,000 images. And we said, this is not going to scale. If we need to produce a machine learning model, we need to figure out a more semi-automatic or automated approach to labeling these, and therefore the need for developing necessary text analytics that can have the kind of accuracies from automated labeling that is comparable to manual labeling. Now, just to tell you the difference based on the technology we developed, we could label about 500,000 images in about 4 days after trying this approach. The actual labeling algorithms itself, they led to research advancements in text analytics, and the approaches we took were initially the machine learning driven, which you saw before, in even generating the vocabulary, but also combined this with the vocabulary driven and language modeling approaches as such. So the basic approach for vocabulary driven is to start from the lexicon that we have and locate these findings and their synonym variants within these images. It also uses a negation detection technology to be able to say that these are negative findings, such as no evidence of certain findings as well, because these are also described in reports. Now, the technology we developed was something that we published in AMIA last year, and you can see that the machine's recognition capability is getting pretty good at recognizing. Now, these are just the core findings that we showed you earlier. And you can see over here, the coverage of the findings is much larger than what is currently the state of the art, which is shown in this blue other colors than the green color. So this is, we cover not only the known set of findings catalog, but also a host of others. And you can see the F1 scores for the detection of these findings in reports. Again, this is something that was published at the American Medical Informatics Association conference. This is the top conference in medical informatics. Now, once we got that, we could describe core finding. But as I said, to produce in the report, we need to be able to describe fine-grained findings. So this means we needed to be able to cover many different descriptors that are associated with findings. Cataloging this itself was an effort, but at a very minimum, we feel there are at least 17 modifiers that describe the findings, referring to anatomy, the subanatomy, the location, the laterality, the severity, and so on. So the next exercise was to see how to do automatically detect how many such fine-grained findings are there. We decided that this problem had to be done in an automated way, and we could not afford to spend a year's time of clinicians to develop as we did for the chest X-ray lexicon. And this was something that was doable with the automated text analytics technology we developed. So to give you briefly how it works, if given an image and it's chest X-ray, you can see sentences like this: clear lungs without evidence of pneumonia. By doing a dependency parse tree analysis of this type of natural language sentence, we can detect, and combining that with the vocabulary driven approach, we can know that some of these terms are referring to diseases or anatomies, like lungs for anatomy, or finding as a clear lung, or negation terms such as without evidence over here. And then once the basic terms are detected, then we use a dependency parse tree, which essentially catalogs the groups, the modifiers that are associated with these core findings, and recursively generates larger and larger phrasal groups to eventually result in a collection of findings and their associated modifiers. So for example, in this sentence, we can catalog that this is an anatomical finding directly from the lexicon. It is a negative finding from the way the negation was detected. And then the finding name itself is a lung of clear lung. It is referring to lung as the anatomy, and so on. And then similarly, we can say from a disease standpoint, there is no evidence for pneumonia which occurs in the lung. So a finding can be described by a fine-grained pattern like this that documents the type of finding, whether it is positive or negative, what the finding itself, and there could be one or more modifiers as they come. So here's another example with a sentence like this: lungs are normally inflated without evidence of focal airspace disease, pleural effusion, or pneumothorax. You actually see multiple findings in one sentence, which we are able to extract and catalog them as such. Now, this work is represents the state of the art in text analytics for finding detection in radiology reports. And this is something that won the Best Paper Award at AMIA conference last year. Now you can see an example from here and how we are able to generate natural language reports from such type of labels. So given an image such as this, the label that is recognized is of this type, and now it is still in a quantized form. However, from here it is easy to generate a sentence which says left basal atelectasis. Similarly, you can see the type of sentences that can be emitted from having a detailed label of this type. And we've done an analysis of this algorithm's accuracy over 5,000 sentences, 5,000 labels and about 3,000 sentences. And you can see that the system is able to do this with less than 4% error. The majority of the errors are still in negation detection, which continues to be a challenge for text analytics in understanding the many ways in which humans express negative findings. And when you do this approach, now mining on the same 220,000 reports, we got over 100,000 labels. Now, this is a challenge for any machine to be able to recognize 100,000 labels from a machine learning model standpoint. So we tried to do a further grouping through this process to get at least 83% coverage of all possible labels found in chest X-rays in a detailed finding level, and going around the 78 labels we have. So this gave rise to about 457 labels. That still represents the largest number of labels ever covered for chest X-rays. Okay, so we have machines ready to now work because we have data and we have labels associated with this, so we can go ahead and try to build a machine learning model. Now, from the way we have described it, you can see that there are a very large number of findings, but of those, even though there are 457 detailed findings we're talking about, the actual number of findings that you will see in a chest X-ray report is 4 or 5 findings. So how do we get to that smaller number, which requires machines to be of very high specificity as well as high sensitivity to make sure that you're not missing any critical findings. So we used a variety of techniques and developed and experimented with several different machine learning architectures. But the basic idea of the type of the first experiments we tried were building specialized models for different sets of findings, such as tubes and lines detection or just a technical assessment, which alone may be of commercial use from the point of view of device vendors who want to detect findings and autocorrect them during imaging itself, for example, or limited use within an emergency setting where if all you wanted to know was whether the tubes and lines were present and if they were placed properly. But we found that when attempting all labels is just it's not as much more difficult than focusing on one label. So our next attempts were to build machine learning models that would cover them all. And here we adopted 3 basic principles of well-known architectures. One was to be able to do a multi-scale description of image features, which is coming from the use of pre-trained networks such as VGG16 and ResNet50. So those who are machine learning audience can relate to these, but these are popular architectures for computer vision. And so we adopted these, but we modified them in a multi-scale pyramid architecture to develop a multi-scale description of images. Then we allowed them to describe this at a each of these findings at different levels of abstraction, as this animation shows, to cover larger and larger areas of the image through this dilated block techniques. And finally, we used a pooling technique to collect all the features into a vector formulation using bilinear pooling, so that we don't lose in dimensionality projection as much before we go to a classifier, which is a standard dense net classifier. You can see the kind of deductions that are done. So even though the machine was a whole image classification machine, it can accurately localize some of these anomalies as well, such as cases of hyperaeration over here. And here's an interesting case of tubes and lines being found in the airway in an APE portable view where the patient is in a prone position, as well as cardiomegaly cases. And here's an interesting case where there is an internal post-surgical material gland, and this happens to be the sternotomy wires around the trachea as such. Okay, so now we come to the last stage of the challenge itself, which is to generate automatically the reports from the machine learning models. As we said, the machine learning model simply spits out a label, but to generate a natural language sentence from this, we need sentence generation technologies. The popular way to do this is through language generation methodologies called transformers, and those that have been following GPT-3 or other transformers that are now automatically writing papers. This is what I'm told is that the modern way of writing papers is have a machine write it for you. Anyway, these methods can write text, all right, can even produce natural language text, but is that meaningful for your chest X-ray? Is it really corresponding to the findings that you're seeing, and it's described in a clinically meaningful way? It's still very difficult for these automated language transformer-based methods. So our approach was to again utilize knowledge from clinicians to guide this process. So starting with a course description such as elevated hemidiaphragm as the core finding, if we can go to the next level and describe a fine-grained finding such as elevated right hemidiaphragm mild, the report can then be generated as the right hemidiaphragm is mildly elevated. So here is where we can leverage actual sentences from reports using a document retrieval paradigm to index from the fine-grained findings that we detect. So an automatic report generation methodology will take a report and extract fine-grained finding labels from there, from which we can basically record in a database the sentences or report that correspond to these findings and pre-rank them for various findings. Now, given any chest X-ray, we actually detect at inference time using our fine-grained finding label generators, and then we index the database to do a document retrieval style retrieval of relevant sentences from reports, which are then used to form a report itself. Now, the report generation ensures that the findings are correct from the way of the detection of the findings, and then the sentences that are correct are ranked both from not only that they describe the finding in the right sense of the finding, but also that they are likely to co-occur in a single report as well, so that they form a plausible description of some finding in some chest X-ray. The overall report generation system uses a dual learner combination of a coarse and deep finding, and then fuses them to do a report retrieval as such. Now, we have published this work at the MICI conference last year. This is the top conference in medical imaging called Medical Image Computing and Computer-Assisted Interaction. So this particular paper evaluated the overall models for 78 findings and for 457 findings. These are the coarse and the fine-grained models, and you can see the AUCs of performance and the kind of findings that can be described by this model, such as moderate pleural effusion in the right lower lobe, which precisely localizes it as well directly from the label. So we also looked at, so here are some examples of automated reports that are now possible to be produced by machines. Some of the demonstration you've seen earlier as well, but this will give you a flavor for, in comparison to ground truth reports, what type of reports can be generated by this algorithm. And we did do a comparative evaluation over different methods in order to get to the types of language generation accuracies using popular measures such as the BLUE score or the Rouge score. But these are lexical matching scores or very weak semantic matching ones. They don't have the power to reflect the clinical significance yet. And so this is still an active area of research, or how to evaluate clinical reports automatically generated by machines. All right, so now we come to the last section of our talk, which is going to talk about how do we validate such models and how do we go and prove that the kind of AI tools that you've developed are really good enough for use in a clinical practice. This is where clinical studies come in. And obviously we need benchmarks to be developed for these as well. But as I said earlier, we also needed to develop comparative performance and record comparative performance on benchmark data sets by radiologists themselves, so we could then assess in a quantitative way as such. So the first type of evaluation, however, we tried to do was to see, is this is good enough for any particular practical use? So the first study we did was to see if the machine can be used to do primarily a normal abnormal discrimination task to see if any of these are abnormal cases or so. So this study was actually performed at the Deccan Hospital in Hyderabad, as I mentioned, and this is a 600-bed hospital, I'm told. And we collected retrospective data for a period of 6 months, and then a prospective data for about 3 months for running, letting them use the service on their own. The definition of normals was to see that their patients came back with no clinical follow-up on and were discharged and not readmitted as such. And it's something that was also validated by a pulmonologist. And the details of the study are described over here. Now, the results from this study, so the training of the model was done on US datasets, but the testing was done on India datasets. And you can see the ROC curve that resulted from our normal abnormal discrimination task. And if you pick a point on this ROC curve, what it is saying is that you can filter as many as 30% of your normals without missing a single abnormal case. And this could be used from a point of view of workflow reduction in clinical workflows. You can imagine a potential use case for this type of study as such. And again, this was published in last year at the ISB conference and is under a journal review. So the next study was to do a discrete label comparison study where we basically collected the performance of radiology residents. Since our goal was to at least be at the level of entry-level radiologists, we decided that the R3 residents in a radiology residency program are comparable for the machine. And we collected over 2,000 chest x-rays or so, and then narrowed it down to about 1998 after some selection process that the radiologist said were qualified images for interpretation. And we actually ground truth these data sets by 3 radiologists through a triple consensus with adjudication validation process so that they could agree on the list of findings actually present in these x-rays. Then we gave about 400 images per radiology resident and asked them to do a discrete read by just selecting these labels. And then we could just do based on that, we can now run the machine for the same set of images and see the comparative performance. Now, these results were described in our JAMA paper last year, and some of you, if you've read it, you can also see the title of this particular paper. So from the results of these were interesting. We have cataloged in a very detailed level the sensitivity, the positive predictive value, and the specificity of per label. And we can also see a relative comparison for which findings are machines about the same level. So you can see for normals, the radiologist and the machine are about the same. And then there are some cases where the machine is outperforming the radiologist, such as the pulmonary edema cases, but there are also cases where the radiologist is much better as well. So in fact, if you see the COPPA scores, you can see the variation like this. And then from the point of view of residents, you can see there is actually variation among performance among residents themselves as such. So the overall result that was significant from this study and this challenge result said that there was no statistically significant difference in sensitivity between the radiologist and the machine, but statistically the positive predictive value and specificity of the machines were better as such. But the overall conclusion was still that the radiologists do much better on difficult findings such as masses and pneumothorax. And if you look at a catalog, so we also say over here which findings are machines about the same, which findings radiologists outperformed, and you can see masses and nodules, and tubes and lines placement is still more difficult for machines as such. But there are cases where the machine actually outperforms radiologists. So the last study in this series was the ultimate study for showing a Turing test for radiology means, as you know, from an AI perspective, a Turing test is considered the epitome of whether the software has reached the capability of a human. And the way it is done is to blind the observer for who is answering a question. It is typically done in a natural language type of task. So for a radiology read, the equivalent was to have a chest x-ray shown and a report that was already produced by unknown source. And we had attending radiologists, which are senior radiologists, assess the report to see whether this is an acceptable quality report. And we use the same grading criteria as the American College of Radiology assessment. And we also allowed them to give a qualitative score from 1 to 10 to see how good they find this particular report. And they had the opportunity to actually document the finding as well. Now, building these kind of interfaces for studies is also an important task that I haven't emphasized, but you can imagine to do a detailed study, there is a lot more involved, even from the point of view of user interface development. So the final result of the study is very interesting. We had the study run live at a conference, and there were 3 radiologists sitting in dark rooms, and one of them got machine reads, one of them got radiologist read, and one of them got a mixture. Um, and so yeah, I'll be done in a few minutes. Sorry. And then the comparison essentially showed that the radiologist read and the machine reads were very comparable in quality, but they were in general more excellent quality reads on the in this particular lot as we go through. So this is still a limited study with about 166 report that could be documented live. We have rolled out an earlier version of this study at RSNA if some of you attended RSNA. Okay, so to summarize, the chest X-ray technology uses you an example of what does it take to develop AI that needs to be primed for readiness of use in clinical workflows, assessing the breadth and depth of the findings itself, but a series of processes that we have to go follow, understanding the radiologist diagnostic process to all the way up to building the machines and developing the associated technologies that go with this as such. And here are some publications if you want to track some of this work in the public domain. I'll be happy to stop here and take any questions.
Thank you. Thank you so much, Dr. Saida, for that fantastic overview of the whole grand challenge, extensive work that you've been doing. If coming from a very healthcare in a perspective, not much into the technical aspect that you've been talking about as such, where do you see this technology coming in a more large scale interpretation, and what are the key innovations or breakthrough that needs to happen for it to be mass rolled out? Where do you see this coming essentially?
So first of all, this is a research project, and although we have done quantitative studies for comparison all, if you want to deploy this in a clinical workflow, you do need regulatory approvals, which is actually going to be the biggest challenge because the current regulatory processes are not designed for large-scale detection of findings. They, with the knowledge that the FDA is using, it has been approving findings, one finding at a time. So if you see the commercial chest X-ray technology right now, there are a couple of vendors who can detect pneumothorax or some very common conditions, at best about 4 or 5 conditions. So to be able to get this kind of wide scale coverage, our first hurdle will be the regulatory hurdle. And I think new policies and new methodologies have to be adopted in the regulatory purview. As you saw from here, it is not the use cases. It is still an assistive use case. So the radiologist is still in charge in terms of overseeing and validating the reports. But the boring process of writing it down as a report itself is something that we can reduce. There are some studies that can be done to see how much reduction in time actually happens. I think there's a recent paper that came out on that as well. But from the point of view of deployment, I think that is one of the first. As far as adoption in clinical workflows, I have shown in the earlier chart, you can deploy it at many steps in your workflow. whether it is at the device vendor level, in next generation technicians can be better trained with AI itself, and they can themselves know whether this is technically assessed is sufficient for interpretation, and the machine can auto detect it and correct itself as well. So the next generation device, when makers can build this into their detection technology right into their imaging equipment itself. But if you go to the priority rate, I mentioned that. So this worklist prioritization tooling is already available, and many AI software are now being used for worklist prioritization to order the reads at a higher level. The more urgent ones go first. And then this type of reporting is something that we think will be expedited, because many times radiology is not available, particularly on weekends and so on. So if right now, what is the choice? The resident is doing the read, and it is as good as the residence quality. So I think having preliminary reads being done by machines is something that will be coming. And it's going to be something, if once we get the regulatory approvals, it will be something that will be favorably seen even by radiology read companies, that where you outsource the reads, because this is not a money-making proposition. A chest X-ray read is like $9 a read or something like that, and if they can reduce the cost to $6 a read by paying appropriately for automated assistance, you can see a savings on the radiology read company. So economically, it may be viable for them. And radiologists themselves are not as interested in reading this because this is a low-hanging fruit for them. The real money makers are the MRIs and CTs. So chest X-rays is not as, from a financial standpoint, as incentive for them. So this is one such domain where you could do this. But the general comment about deploying these in workflows, you have to prove the performance through studies like this. This is something we learned during our early experiments with RSNA community, that publications is very important for documenting the performance of these. But the second thing is the breadth of coverage is also important. There were some questions in the chat.
It's very exciting. Yeah, if you could, you want me to speak to it, or you could take it either way.
No, I can see those. I can answer a few of those, but I want to be sensitive to your time because we are a little bit over.
I mean, we are fine with it, essentially.
It says, can AI help in controlling, preventing? Okay. I will have to step out soon, but let me answer a few of these questions. So it says, can AI help in controlling or prevention of the disease? So this is a predictive use that they're talking about. Now, there is work going on in predictive analytics using AI. And if you have past records that have documented these findings, and particularly in the COVID-19 case, there's now a lot of data becoming available. So one can use AI in a predictive way, yes, very much so. Now, in that sense, it can help in anticipation and in use in future. As far as controlling the disease spread, that is much more than just AI. There is a lot of other decision making involved, and measures that the government can take, and the institutions can take, and people themselves can take in helping prevent the spread of the disease. So I would say that AI can help, and even there is tracking devices that tell you how the disease is spreading. So AI can be used in many ways, even in those situations, but ultimately whether that can guarantee that the device will, the disease will not spread is a function of many other factors. Can this technique be used for identifying lungs infected with COVID-19? Okay, now, interestingly, when COVID-19 came along, all the imaging people jumped in the field to see, hey, we can go ahead and detect this. It turns out that the detection is not that difficult. And in CT, it is pretty, easy to detect. So we found that the actual detection of COVID-19 is not as an interesting research problem, because this is relatively easy, but for machines. But I think the more important problem is, what is the what is the progression of the disease, and what would be the outcome for such patients. And this is still a challenge, because as you saw, different people have been affected by the disease to different extents. Some people just take it in their stride, some people get hospitalized, Some people recover from these. So what is making different people react differently, and studying that is more than just imaging alone. Imaging can be one of the cues, but you have to augment it with other information at the genomic level, at the clinical factors level, so a host of other things. And that's where the active research is now, is combining and doing a fusion of multimodality cues to do better prediction from a disease progression standpoint.
Can I ask a question, Zaida?
Yeah, go ahead.
Well, it's not a question, it's such not on a personal note. I was reading on the IBM site on your research, and it shows that your journey into radiology assistant work that you've been doing is started with your father's misdiagnosis of stroke. Has that still been a passionate driver for you working in that field, essentially?
Yes, I think many times if you want to work long term in a field, you have to be passionate about it, and you have to care about, the outcomes that you would like to see the change. So the way I started in the field of clinical decision support was indeed to guided by that incident and a desire to change what happens at an emergency room these days when you either due to lack of right clinician being present, or simply due to the pressures of what happens in a high volume setting. Errors are being made, and I think this is an acknowledgement that we are all humans, so these errors do get made, and improving and having machines to assist them is a valid use of artificial intelligence, particularly in healthcare. So yes, I'm still guided by these, and even now the new projects that have started are looking at improving the lives of humans and by machines. That
Fantastic.
is still the
So
goal.
it's for a country like us in India, essentially, where obviously there are a lot of shortage in terms of skilled manpower, like radiologists. And if you look at per 1,000 people, do you see a significant impact of this work in countries like India as such?
Yeah, very much so. In fact, the way we were running this service, the normal abnormal discrimination service at the Deccan Hospital, the planned usage was they could simply upload this image to the service and they get back a report. And then, and we did this obviously in a parallel workflow setting, but the reaction from the investigators working with this was that the machine was, they began to trust the machine in terms of the data that was coming in. So if this sort of performance, so the key to this is good performance. If the machines are able to put a reliable performance that engenders trust, then one can see this being more widely deployed. I mean, how many people, and in US use automated driving right now with Teslas and all. So people are beginning to trust machines, but one single incident or other can, yes, can damage the reputation, like it happened for Tesla for some accidents. But these are the hiccups during the development of the technology. But over a period of time, I think the overall good, the betterment of the community will come in place, and this we have to continue doing these type of studies with broader and broader applicability. And if there are any institutions in India who want to try or want to engage in these type of studies, we'll be happy to entertain some of them.
Fantastic. Thank you so much. My brother is a radiologist, a pediatric radiologist at Osmania in Hyderabad. And I was just talking to him the other day, and he was very fascinated about the whole impact of, augmenting, radiologist role by AI, essentially. I think we don't have any other further questions.
Yes.
And I think we're all running out of short of time also.
Okay.
You've been always here for more than an hour or so. So thank you once again, Dr. Saida, for this fantastic talk, and it was really enlightening for all of us. And just for your information, we have more of them from the healthcare domain here. Almost 70% of them are the faculty from the pharmacy as such, and 30% are engineers out here. So a lot of interest within that pharmacy community about the role of AI, and it was really refreshing to hear the cutting edge work that you've been doing at IBM. So once again, on behalf of all of us here, faculty who have come all over from India, I really like to thank you for your valuable time. It was a lot of quite learning, and definitely we would like to continue this conversation with you and in different areas as such. It could be joint collaboration, or it could be more of a as you mentioned, with Edison interdisciplinary work within institutions. So we'll definitely get back to you on that, and we'll try to explore what can be done. As such, you're already working with Deccan Hospital. Maybe we can start this, a similar venture here in Mumbai as well. So with that,
Okay.
thank you once again for your
Thank you
time.
very much.
Really, thank you. And then hopefully we'll have again, in future we'll have you here again for sharing some of your more updated work on this Grand Challenge project.
Yeah, maybe another grand challenge several years down the line, but it was great. It was a pleasure for me to
Thank
talk
you.
to your community as well, and I hope this has been a good learning experience.
Absolutely.
Thank you very much for your time.
Thank you much. Thank you so much, ma'am. Thank you. So moving on to our next speaker out here, we have Dr. Richa Mishra here, who's coming in from the Computer Science Department at Vishwaniketan Institute of Technology, and she's been extensively working in the areas of strategic consulting and use of technology asset, and she has a lot of passion, 13 plus years in teaching and in-depth academic research, PhD in computer science and engineering. She has a special inclination for bioinformatics and health informatics. So with 2 international patents, Australian and 4 filed Indian patents, she's an avid thinker along with teaching consciousness. Interestingly, she's the only speaker invited at the 18th Annual World Congress Award Congress at the Los Angeles Convention Center in California, 8th of July this year. Hopefully she'll be able to travel there. And she's also pursuing short-term PhD post-pilot research project at the Deccan University in Australia. She's been recently appointed as senior member of Iranian Neuroscience Society and received research fellowship for the duration of 2 years at the Dana Brain Health Institute in Iran. So without further ado, I would like to welcome Dr. Richa Mishra here for her talk. Dr. Mishra, you have permissions to present. Please check at your end if you're able to do that.
Yes, sir.
Yes, madam. Great. Okay. You can see it.
Is it visible?
Absolutely, yes. Great. Okay.
Yes. Good evening, everyone. I am Dr. Richa Mishra. Would like to talk on the protein structure prediction in autistic children with machine learning. I am not from the bio side, so kindly excuse me if I do something wrong. I'm from computer science and engineering background, and working from last 6 years with autistic children and the problem related to neuro developmental side. So when I see in India, basically we are just in the psychology side. When I see things like it's still more on the behavioral side. So when I found this flaw, there is a gap that as a computer engineer, we can put forward few things and give the proper prediction for the drug development also, rather for the good practices also. So I have jumped into this field and now I'm doing 2 projects. So I'm just here to brief you up about this topic. Now I'll take a quick, make you see what quickly we are going to cover in this, because already I am short with little time. So I'll be starting with bioinformatics and the technology and skill sets we need to pursue our projects into this side. Then the protein and protein analysis. Apart from that, the computer-aided drug design, and then the proposed model for which I am doing for my project. So this is my outline. So now I'm just would like to explain what is bioinformatics. I just say that there is nothing which we can keep separate, the biology and computers that go on and on to explore the new world. So from the interaction of species and population to the function of tissues, cells within an individual organisms, biology is defined as a study of living things. Biologists were used to collect and interpret the data. So what we need in this century, like more sophisticated laboratory technology that allow us to collect data faster than what we can interpret. And we have a vast volumes of DNA sequences data at our fingerprints. So now what we need exactly is how we can figure out, or like how we can predict for a better modeling. So this is actually the need of the hour, the area of the concern, that we have lot of content, but we don't know how exactly this will function and give us proper positioning, and how do we predict what protein will look like based on the knowledge of its sequences. So we need to understand relatively simple code that translates DNA into protein, but how do we find meaningful new words in the code and add them to the DNA protein dictionary? So this is the major task. So now what is done in bioinformatics? So bioinformatics is a science of using information to understand biology. It's a tool we can use to help answer our questions by doing analysis and interpretation, development of new algorithm and stats. So there is a point that whatever the previous algorithms we have, we can utilize the part of it, along with we can develop a new concepts, and we can just figure out whether the accuracy is there with our new model or not. And the third part is we can develop and implement the tools what we have, the database we have, to figure out exactly what exactly we are looking for the output. So we can say in these days, this is the buzzword status. The terms in being used in a number of ways depending on who is using it. Strictly speaking, bioinformatics is a subset of the larger field of computational biology, the application of quantitative analytical techniques of modeling biology systems. So I must say the field of informatics relies heavily on the work by the experts in the stat side and the pattern recognition. Actually, there is not a magic anywhere. Like we just find, we just want the result side. Just a second. Hello, am I audible?
Yes, ma'am, you're perfect.
I don't know, sir. My screen is stuck. I don't know.
We can see your screen here. It says the one which is bioinformatics important.
Oh God, just a second, sir.
You might want to stop sharing and read.
Just a second, sir. Actually, my screen is stuck. I don't know. It's not.
Now it is going to the next slide. It says bioinformatics skill set.
Yeah. Okay. So actually, the bioinformatics people, or the bio side, is looking for the prediction, but there is actually not any magic we can expect. It is having a lot of pitfalls for those who look for the patterns and make predictions without a complete understanding of where the biological data comes from and what it means. So when I started with the project, I also was struggling with exactly what I'm looking for, or what type of prediction I should do, what is the protein sequence, everything. So I had to start from the scratch, because without understanding the part of it, you cannot do the prediction modeling and the result analysis. So provided we should be aware of the complete sequence of or I can say the tools of bioinformatics makes it possible to do exciting things, like we can compare DNA sequence, can generate results that are potentially significant. When I say potentially significant, it is perhaps the most important phrase. These new tools also give you the opportunity to interpret data and assign meaning where none of these actually exist beforehand. So where I am now in my field, I have really shortfalls of the previous data, along with I don't have much of the literature part even to study and do the prediction modeling. Like in case of India, we don't have that much even the database, and the conclusions or whatever the study is available is mostly from the outside world. So this is challenging. So before we start doing with any projects related to bioinformatics, we should have the skill sets of stats, So because there is lots of data and lots of missing values is also there. So we need to understand how we are going to fill that part. The programming languages, mostly the Perl and Python is good to go. This is my experience, says like scripting languages can go with Python, Perl and Ruby. I have experience of Perl and Python, it's good to go. And extensive use of text file formats needs parsers. We need to understand the compiler side and the talk subject. So that helps us to do more deep study about it. And the integration of all the tools along with that, we should be aware of the databases and the data structures and the modeling. For modeling, we have the domains available like discrete or continuous domains, the scientific computation packages. We can have the modeling with the help of R programming, MATLAB and all. Apart from that, we should have a communication skills. Why it is very needed? I've just taken one example. Why we are looking for, if someone from the bio side wants to present to a computer scientist or a mathematician that, okay, I want this to be done. Okay, so if someone goes with the question that, okay, I just want to understand the effects of the gene X with the condition Y, I would like to know the consequences and the result. So point is, as a computer engineer is a layman and doesn't know exactly what to do, just they need to understand the input and the output of the problem. So point is, communication skills should be very clear between the two people, those who are from the computer and from the bio side, and then they can take it ahead. So it is very important to understand in which triangle you belong to. Are you from the biology and the medicine side, the computer side, or a bioinformatics or mathematics and a stat side? If you are able to figure out what exactly or where you belong in the triangle, then it is very efficiently you can club and can figure out how you can take it your project further. So just an example, it is just like as we start doing the project, it's like whenever we are trying to figure out anything, we find, okay, everything is the result. So this is not done. So someone with the expertise domain should be there to help out the results what we have, and the authentication procedure should be properly done. So this is what I personally feel. Apart from that, if we do have the knowledge domain where we can apply all these in the field of medical applications, like to find out the disease or how healthy we can go ahead in our lives, and the pharmaceutical and biotech industries to develop a new and a better drugs to do with gene-based or the structure-based drug design, and the agricultural application sides also we can apply this information. If I see where this technology is going parallel with biology side, so I can see it's like going on and on together. Like, and you can see the organism's hereditary and functional information is stored as DNA, RNA, and proteins. So all of which are linear chains composed of smaller molecules, and these macromolecules are assembled from fixed alphabet of well understood chemicals. So exactly, before I go into the different parts, I just would like to make you understand this presently, this particular diagram makes us understand that like in the part of bioinformatics, sequence comparison, database searching, sequence like designing, and the populating database. All these are very, very complicated part, and the bioinformations go beyond just capturing, managing, and presenting data, drawing inspiration from a wide variety of quantitative fields, including stats, physics, computer science, and engineering. So particularly this diagram shows us that we go on and all together, and the main goal of bioinformatics isn't developing the most elegant algorithm or most, you can say, arcane analysis. The goal is of finding out how living things work, basically like the molecular biology method that greatly expanded what biologists were capable of studying. Bioinformatics is a tool and not an end in itself. So point is, you can see with the diagram, you can see we have stats, we have the sequencing part, the computational ecology, the high-tech field ecology, all that along with data standards, everything is like interlinked. Apart from that, we need mathematicians, physical modeling, the computation part, the information technology, and the sequence knowledge. So everything is interrelated. We are looking for it, and that's what we cannot say that, again, it's a stuck. So it's like bioinformaticians are the tool builders, and it's critical that they understand biological problem as well as the computational solution in order to produce useful tools. So here, what I can say, the team should be of different domains and different expertise so that project goes well. So what type of information basically is mean to biologists? What exactly is the information for them? Okay, so if we have a lot of sequences or a lot of information in the form of representation, organization, the manipulation part, the distribution and maintenance of the use of information, particularly everything which is needed is like in the form of digital, so that the interpretation part should go well, and quite possible to go out and apply for, this particular knowledge domain into variety of things. So developing analytical tool to discover knowledge in data is the second, and more scientific aspects of bioinformatics, they are mainly level at which use of biological information, whether we are comparing sequence to develop a hypothesis. So point is, when we start doing a project, we do make a hypothesis about a function of newly discovered gene, breaking down known 3D protein structure into bits to find patterns that can help predict how the protein folds, or modeling how proteins and metabolics in the cell work together to make the cell function. So the ultimate goal of analytical bioinformaticians is to develop predictive method that allows scientists to model the function and phenotype of the organisms based only on its genome sequence. So what is the goal basically is to divide the whole part into a small chunk and go all together. So point is, what is a challenge as if now? If I see cracking the genome code is complex, and at the very simplest level, we still have a difficulty identifying unknown genes by computer analysis of genomic sequence. So we still have not managed to predict or model how a chain of amino acids fold into the specific structure of a functional protein. So in my case also, if I'm from a computer side and I am just trying to figure out what exactly the protein sequence, I exactly need to understand, because the previous results are not there to compare. So this is the flaw and still a part of study. So this, I can say, if we are into the field of bioinformatics, we are looking for the more global perspective, because as there is a lot of data, but that data should be used in what way, or to get into what kind of prediction. And the machine learning and the data mining, exactly how we are going to make our testable hypothesis, these are very important aspects to understand. So what are the units of information do we deal with in bioinformatics? DNA, RNA, and protein. Now, I would like to talk about biology premier. So what is life made of? Simply, we all are aware of this. I'm not going to take in detail, but I would like to take this point, because this is actually the insights of, or you can say the beginning of any project. So what is life made of, and where does the variation in genome come from? So it is very important that we should talk about the cell. So except for viruses, all the life on this planet is based upon the cells. Typically range in the size of 2 into 10 to the power minus 6 meter to 10 into 20 into 10 to the minus 6 in diameter. So cells sequester biochemical reaction from the environment, maintain biochemical component at high concentrations. So this is like the unit, you can say, for every living system. And there are two parts there are two different types of cells, pyrokaryotic and uracotic. Okay, so it's like instead the DNA is condensed into the nucleoid, which is less highly structured than you let chromosomes and appear as disorganized ball. Okay, so we get to understand these two parts, and then three critical molecules is like the major part of all life. Like one is DNA, RNA, and proteins. So if you want to explain what is DNA or RNA or a protein, so it is important to understand, because DNA is going to made up of 4 deoxyribonucleotides like adenine, thymine, cytosine, and guanine. And RNA is made up of 4 ribonucleotides, that's adenine, uracil, cytosine, and guanine. So point is protein. I need to talk about protein. Protein are made up of 20 amino acids. Because of these macromolecules are linear chains of defined component. They can be presented as the sequence of symbols. So these sequence can be compared to find similarities that suggest the molecules are related by form or function. So very important to understand the DNA, RNA, and protein. So simply the DNA, the code of life. I'm not going to talk about the discovery of DNA and all. This flow of information we need to understand from DNA to RNA and then RNA to protein. And if we understand the protein structure, that gives us the clarity that exactly what we are looking for, or what type of symmetry we are, or the similarities we are looking for, so that we can plan our prediction modeling. So sequence comparison is possibly the most useful computational tool to emerge for molecular biologists. So the World Wide Web has made it possible for a single public database of genome sequence data to provide services through a uniform interface to a worldwide community of users, with a commonly used computer program called FS Blast. So in my project also I have used FS Blast only. A molecular biologist can compare DNA sequence to the entire publicly held collection of DNA sequence. So now, so I've just said protein is a collection of 20-letter alphabets and amino acids. Let's talk about the amino acids. So these are the building blocks. Like protein, what protein is, like deciphering the 3-dimensional structure of protein from its amino acid sequence is a long-standing goal in a molecular and computational biology. The building blocks is like protein sequence consists of 20 different kinds of chemical compound known as amino acids, and they serve as a building block of protein. Amino acids contain a central carbon atom C, which is attached as an hydrogen atom, an amino group. So what we need to understand about the amino acids, because they are naturally of 20 different types as specified by a genetic code emerged from DNA sequence. Furthermore, what we can say, non-natural amino acids occur the rare cases, and this is we need to understand in a better way, as the products of enzymatic modifications after translocation. The major difference among the 20 amino acids is a side chain attached to the C through the 4th valence. So, so now we have to understand the analysis of protein and nucleic acid sequences. So for that, we need to get into the parts of protein, the different structures. So genetic information is stored in DNA presence in the nucleus and transferred from one generation to the other generation. So DNA transfers the information to the messenger RNA by the process of transcription. So the correct transfer of the information is ensured by the complementary base pairing between the nucleotide present on the DNA and mRNA. So the mRNA transfer this information in the form of protein by the process of translation. So DNA is made up of 4 different types of nucleotide, that's A, T, G and C, and the triplet of nucleotides codes is responsible for coding for amino acid present in the protein. So when we are planning for a coding properly, then we need to understand all the sequence before we enter into any machine learning process. So this is very important. That's why I took this point, like how we are going to analyze the protein sequence. So it is like it is made of different types of amino acids, and the composition of protein is determined by the DNA sequence. So here in this diagram, you can simply see that the sequence of nucleotide bases as well as amino acid sequence of the protein has wealth of information used to understand the structure and the function of macromolecule. In this, we will discuss how this conclusion will be drawn when I'll take an example of my own project. And now let's talk on the, I don't know why this is happening. Sorry. Let me talk about the structure of nucleic acids. Nucleotide, the building block of nucleic acid, consists of pentose sugar, base, and the phosphoric acid residue. The nucleotides are connected by a covalent linkage between the pentose sugar of nucleotide and phosphoric acid of the next nucleotide. So in this diagram, you can simply see there are 5 different types of nucleobase attached to the sugar through a N-glycoside linkage. Uracil is found in RNA where thymine is present in the DNA. So these nucleotides are abbreviated with the first letter of the base to write the nucleotide sequence of the nucleic acid, such as adenine is denoted as A. The base have a specificity towards the other base to form a pair through hydrogen bonding. A is making 2 hydrogen bonding to the T, whereas G is making 3 hydrogen bonding to the C. So DNA is a double helix structure with the bases presence on both strand, and the sequence information on one strand of DNA can determine the sequence of the other strand. So this is about the structure of nucleic acid. Now see the structure of the protein. So the next side diagram, you can have a look on it. The protein is made up of 20 naturally occurring amino acids. Typical amino acid contains amino and a carboxyl group attached to the central alpha carbon atom. So in this diagram, you can have a look. The side chain attached to the alpha central carbon atom determines the chemical nature of different amino acids. Peptide bonds connect individual amino acids in a polypeptide chain. Each amino acid is linked to the neighboring amino acid through an acid amide bond between carboxyl group and the amino group of the next amino acid. So every polypeptide chain has a free N and C terminal. So in this figure, you can have a look. The primary structure of a protein is determined, or you can say defined, as the amino acid sequence from N to the C terminus with the length of several hundred amino acids. So the ordered folding of polypeptide chain gives the rise of 3D conformation known as secondary structure of the protein, such as helices. So we should be aware of all the type of structuring of the protein. Okay, so this gives us gives us the rise of 3D conformation, which is called as your secondary structure of protein, such as helices. You can call it a sheet and loops. So arrangement of the secondary structure gives rise to the tertiary structure. So alpha helix and the beta sheet are connected via unstructured loops to arrange themselves in the protein structure, and it allows the secondary structure to change their direction. So tertiary structure defines the function of the protein, enzymatic activity, or a nature of a structural protein. So different polypeptide chains are arranged to give tertiary structure. So in this, it is quite clear that how this structuring is. Next, I would like to make you see how is your your 4 types or different levels of organization in protein structure. So you can have a look. There is a primary structure, then it goes to a secondary structure, then tertiary, and then finally the quaternary structure. So now we should know about, after the structuring part, we should be aware of what is the biological databases we have. So there are primary database, there is a database of protein sequences, secondary database, the prints and molecular structure database. So I will take just a quick way to make you understand what are these biological databases, because we will be needing it further. So the biological database is in the post-genomic era, nucleotide and the protein sequence from the different organisms are available. So point is, we are not going to figure out exactly there is a depository. We can just go and have a look on a previous sequences so that we can compare exactly what is now the new finding. So it has paved the determination of the secondary and the 3D structure of protein as well. So this vast amount of information is processed and arranged systematically in different biological databases. So the information present in these databases can be used to derive common features of the sequence class and the classification of an unknown sequence. So in a primary database, this is what the collection of the data obtained from the experiment, such as a sequence of DNA. So when we start doing any of the project, we start with the primary database. What is that? Because as it is obtained from the experimental side. The sequence is obtained of DNA or a protein or a 3D structure of a protein. So once we start doing our project, we would like to start with the primary database only. And the other database of the nucleic acid sequence is GenBank. The other is Entris, EMBL, DDBJ. These are few more databases available for nucleic acid sequences. These are first, GenBank is a public sequence database. You can directly go and access. I have given even the link site, ncbi.nlm.nih.gov. So you can directly go and have a look. Exactly, it is quite simple to access. You can go and directly use for your projects directly, even if you want to start doing the machine learning. So you can just go and take the data and can start working on it. So the entry into the GenBank is made through a login into a database with a prerequisite of publication of a few sequence in any scientific journal. This is important. Whenever you are into any project or you have done any sequence, the scientific work, then only you can go and just log in and start working on it. So each entry in the database has a unique session number and it remains unchanged. So for an example, a sample GenBank entry can be accessed via a link, which I have shown you. So a typical GenBank entry has the information about the locus name, the length of the sequence, the type of the molecule, DNA and the RNA, and the nucleotide sequence of the entry. The other is entrance. It is used to search all NCBI association database. It is a powerful tool to perform simple and complicated searches by combining keyword with the logical operator, that's and not. So for an example, if you want to search a protein kinase sequence in a human, can be done by, like for an example, with the syntax like Homo sapiens, like ORGN and protein kinase. So simply by putting all such things, you can simply find out the sequence. Okay, so EMBL and DDBJ, that's the nucleotide sequence database presents at European Bioinformatics Institutes. So once you want to use it, you have to take the permission and can access by the link. So that DDBJ is a DNA sequence database present at the Center for Information Biology, like Japan. So there, DDBJ can be accessed by the link which I have shared. And every day the GenBank and EMBL and DDBJ synchronize their nucleotide sequence as a result of searching of a nucleotide in any of the database is sufficient. So they're interlinked and they keep updating their database. And the other is database of protein sequence, that's Swissport NCBI protein database. So whenever you want to enter into the field of bioinformatics, you categorically go to NCBI protein database, and from there you can get the Swissport PIR database, PDB database, and other known databases. So once you want to start with any project, you can directly go to NCBI protein database, and that's the detailing you will find very easily. The other is Uniport. It's like Georgetown University has came together and collected the protein information in the form of centralized catalog known as Universal Protein Resources, like UniProt. So it contains the information about the 3D structure, expression profile, secondary structure, and biochemical function of the protein. So UniProt consists of 3 parts: the UniProt knowledge database, that's UniProt KB, UniProt reference, that's UniRef, and UniProt archive, that's ARC. So these are like collection of Swissport and Tremble database only. So UniRef is the non-redundant sequence database, and it can allow to search similar sequences. UniRef 100, UniRef 90, and UniRef 50 are the 3 different versions of the database, allow searching of sequences, that's 100%, which is less than 90%, and less than 50% identical to the query sequence. So it is important, if you want to directly jump into Uniport and subside the other part, you can do that part. Then you have a secondary database. Okay, so the analysis of the primary data gives rise to the development of secondary database. Once you have completed your first phase of the project, you will be having then your secondary database. Then the secondary structure, hydrophobicity, plot, and domains are present in the various secondary databases. So now we look for ProCite. That's one of the secondary biological database which contains motifs to classify the unknown sequence into the protein family or a class of enzyme. It can be accessed with the web address of http://procite.expacy.org. So the database, which contains motifs derived from the multiple sequence alignment, the cured sequence is aligned against the multiple sequence alignment to determine the presence or the absence of motifs. So once we are doing that project where we are looking for the presence of motifs, so we can directly go for ProCites and look for the sequences. So typical expression in the ProCites has the 7 amino acid positions. Like first position can be E, F, T, N, or A, and second for HFDAS, third for HYT, or fourth can be amino acid except ADS. Okay, and fifth and sixth position by any amino acids can follow, and the 7th position will be proline. So a query sequence can be analyzed using the algorithm scan ProCite. In addition, it may allow to search the sequence with similar pattern in Swissport, Tremble, and the PDB databases. We have further prints and PFAM. So that contains the profiles of protein sequences and classifies the protein families as per the overall profiling. So the profiling is a pattern of amino acids in a protein sequence and determine probability of a given amino acid. So PFAM is based on the sequence alignment. High quality sequence alignment gives the idea about the probability of the appearance of the amino acid at a particular position and contain evolutionary related sequences. So once in few cases, if you see a sequence alignment may have the sequences with no evolutionary relationship to each other. Point is, when we are trying to start a new project and we don't have any previous, like in my case, I'm also struggling with the previous results or the databases. In this critical analysis of results from the PFAM database is necessary to draw conclusions. So when you are using this, you should have the final conclusions because there is a lack of previous collection of the database. So there is InterPro, that's like Swissport, Trimble, ProCite, PFAM, Print, ProDom, Smart, and TagFrams are integrated into a comprehensive signature database known as InterPro. So the result of this gives the output from the individual databases and allow user to compare the output considering the algorithm used in each database. So I'll not go into detail, but directly I will be telling the molecular structure database. We have PDB. This is mostly used when we are writing any paper, like research paper. Mostly we do go and get the sequences from PDB for majorly the comparison part. The way we get the results at our level, and if we need to compare or we need to do the accuracy check, we should have the database to go and cross check. That's we can get it from the protein data bank, that's PDB. Then we have a SCOOP, that's a structural classification of the protein, and PDB gives us a collection of experimentally determined crystal structure. So that's also a very, very important thing to go with. And CATH is simpler to SCOOP, classifies the protein into 4 categories like class C, class A, topology T, and homologous superfamily H. Okay, so now look for the sequence comparison. After this, to know our databases and what we are looking for or what type of inputs we have, then we need to plan our sequence comparison. This is just, I figured out all the major biological data and information sources. If directly you want that these are the subject areas, these are the sources we have, and this is a link. So I'm going to share all this presentation with you all, so you can directly put forward what type of subject you have, what source you should go into, and there is a link associated with it. Then let's talk about sequence comparison. For that, we should be aware of, or we should know what is homologs. Homologs are like 2 related sequences which are termed as homologs to each other. These can be either orthologs or paralogs. The homologs protein from 2 different organisms with similar functions are termed as orthologs, where the homologs protein with different proteins with different functions in organisms is called paralogs. So we should be aware of these 2 types. The homologs is like 2 genes, GB and GC, evolved from the same ancestors, and we look for, these are what called as homologs, and this we need when we are planning for sequence comparisons. So this is very, very important part. Okay, and then why? Because we have to look for identity and the similarity level. So these are the 2 parts, the orthologs and paralogs, okay, which I have just now told. The orthologs, like where the homogeneous protein with different proteins with different functions in organisms are called paralogs, and the homogeneous proteins from 2 different organisms with similar functions are termed as ortholog. So now, what is needed, like you should be aware of, like when we have to plan for protein sequence analysis, we have to do a sequence alignment. So this has to be done with the functionality of identity and similarity. So what is sequence alignment? Okay, sequence alignment. So the analysis of protein sequences provides the information about the preferences of amino acids and residues and their distribution along the sequence of for understanding the secondary and tertiary structure of protein and their function. So the identification of similar motifs in protein sequence would help to predict the structurally or functionally important regions. So the profile obtains with a single amino acid properties based on amino acid sequence would reveal the clustering of amino acids with similar properties. So when we go for machine learning, we have to understand the clustering part. Clustering means the same sequence make a cluster, the area where they shows. So once we insert the data, we look for the clustering part. So the profiles obtained with a single amino acid properties based on amino acid sequence would reveal the clustering of amino acids with the similar property. So we need to understand which are the similar properties should have one cluster. Furthermore, the comparison of different amino acid sequences using alignment method would enhance our knowledge about the availability of similar sequences, and these sequences could be used as a template for protein 3-dimensional structure prediction. So this is very important when you are planning for protein prediction modeling. You should understand basically about this, the protein sequence sequence alignment. Okay, so the comparison of 2 protein is similar, mainly carried out by aligning the sequence of the structure. So in this method, 1-to-1 correspondence is set up between the residues of 2 proteins So the simplest observation is the global alignment of two sequences in which the two proteins have maintained a corresponding over the entire length. So which is important. So with this diagram, I will explain you further. So just have a look with the alignment of two sequences. We have A, we have B, but they should obey the following conditions. What are these? First condition is all residues should be used in the alignment and all should be of in same order. This is rule number 1. The rule number 2 is align one residue and A with another from B. This is rule number 2. The rule number 3, a residue can be aligned with a blank. Okay. And for the fourth one is two blanks cannot be aligned. So these are the rules given to do the alignment of two sequences. Okay, so different ways of aligning two sequences. We have VEIT, GEIST, okay, and PRET, ERIT, which is shown here. You can have a look at alignment 1, 2, and 3. So these alignments, one could estimate the score. What is score? I'll explain you in the next slide itself. What is the score? We need to understand, if there is a blank, there is no score. For everything, for every bit by bit sequence, we have to give the ordering part, and then we have to calculate its score. So we need to estimate the score for each aligned position and hence the total score. So it is important. So the scoring scheme is like score for like let's suppose we are taking score equals to 1 when if both the residues in the same position of sequences A and B are same. So when we are looking for the positioning, if they are at the same place, then we are going to put a score 1. In alignment 1, both the sequences A and B at position 3 are E. And hence what we can say, we have a score of 1. Second part, if the residues are different, that's like score 0. So position, just take an example, like position 1, the residues are V and P respectively, and what we have in the sequence of A and B. So this is score 0. How we are going to do it? When we are finding the residue score are different. The third is when the score is -1. So if there is a gap in the alignment, let's suppose position 2 and 4 in the alignment 1, the added score for all the residue gives the net score for the aligned sequence and the alignment, the positioning of residues with the similar properties. So these are things which we are going to use to find the similar sequences. These you can go and read in detail from the further explanation in Hammer paper that's in the available very easily, EID Hammer. If you type that, you will find the detailing, because I had to also understand this part, because that's not directly shown in any of the book very easily. That's a simple example I could get to how you're going to calculate the score. And this is very important to understand, because we need to decrypt the structure before we start predicting. Okay.
Dr. Mishra, just sorry to interrupt. We have another 5 to 7 minutes remaining. We're running out of time.
Yeah. So these are some programs for aligning sequences. I'm not going to talk in detail about these. So the other is we need to understand the sequence alignment. There are like biological problems we should address, because if we are predicting some drug or like any problem related to health that's come under the category of biological problem, then we have to understand 3 types of alignment: the global alignment, the local alignment, and multiple alignment. And to understand the sequence alignment, I've just taken an example where there is a similarity. You need to find, or you have to calculate the length sequence, and you have to understand whether there are the matches. So just see in the second diagram, there are 2 matches, 5 mismatches, and 1 not aligned because there is nothing. So it's like shown with the dash. Okay. So these are the few things. Then we talk about the identity and similarity. It is like we need to find out the ratio of identical amino acids residues of the total number of amino acids present in the entire length. Then the sequence alignment of nucleotide sequence, how we are going to calculate the score. These are the procedure where we can put into matrix and we need to calculate the 1 and the number of zeros. And finally, we need to calculate with the help of, it's like blue sum 62 matrix. Okay, then we need to understand the score size. The maximum the score, it is good in practice. So this is a local alignment, the global alignment for the sequence, and this is for the protein sequence. This is the study. These are some of the tools which I have figured out about, and where we can use computer for the drug designing. That's very good aspect to talk about, like in this area where we should have the knowledge of computation along with the knowledge of algorithms, and moreover we can find the proper sequencing. So this drug designing and the discovery is a long process. It takes a lot of time. We should know what exactly we are looking for, the what type of target for the drug screening and selection of the inhibitor, toxicity analysis, and lot of analysis related to pharmacological or and where after the prediction are done or the designing done, how we are going to use it for the therapeutic purpose. So the whole purpose or the process of drug design and discovery through the traditional trial and error approach is a very lengthy and a time consuming, but when you're using the computer into it, it is quite reliable and good to go. And now for these, there is a step like structural determination of the target enzyme that can be done with experimental method, which is very like usable. But now where I'm going to use the modeling apart from this experimental method, the other is homology modeling. And these are the steps, like first step is identification of a suitable target, then we need to find out which target and the protein sequence, then the model building, and then energy minimization and the structure validation. There are these points I'm not going to take. These are the some steps for structural modeling, and these are the tools. Now, this is what my project is all about. Autism spectrum disorders are the group of commonly occurring now these days. You must not have heard about autistic or autism or ASD. These are the 5 major problems come under the umbrella of ASD, and these are now increasing from the last 10 years. It's like in every 10 children mostly there were like one were affected, but now it's like increasing, the percentage is increasing now. So you can see that there is a high demand that we should have the proper diagnosis with the help of the genomics and the protein side. So this is the work done, the human gene C3ORF58 or deleted in autism, that's DIA1. Both gene products encode signal peptides for targeting to the secretory pathway. So evolutionary medicine has to be figured out. So this is the project, and for that the part is like protein structure prediction modeling is taken care with the help of dynamic programming along with the k nearest classification. So this is what my proposed work, or you can say this is my ongoing work. In this modeling, I'm using the machine learning part. The machine learning model is giving with the help of like AI. In this, it's giving us the concept that the systems can learn from data, identify the patterns, and making decisions with little or no human intervention. Because before we enter into the final human intervention, we should figure out exactly what is going to happen. So this data analysis technique enable the automation of analytical model building. So there is sequence alignment. We have to understand the sequence alignment, the structure alignment, along with the dynamic programming and k nearest neighbor classification. So this combination is giving the new modeling part, which I'm using. And then this deploy the machine learning to recognize the structural environment of a known homology and their models. So I'm focusing mainly on the alignment generation. So this is all about the detail of how we are going to score. This is global alignment, dynamic programming, what it is, and the rules, how we are going to fill the metrics. And this is the automation. This is the input I have taken and how it has been projected. This is with the help of the talk knowledge, the theory of computation, how we are going to utilize it to finally convert into the modeling, 3D structuring. So this is finally which we have to include or which we have to exclude, which to find out the similarity in the proteins. So this is, and this is the final result I got. And what I can say finally is, it's giving, like when I study this DIA1 family proteins, it is giving the critical role in protein structure and giving us the better understanding about the autism and mental retardation. So this study is making me understand the difference of the genes of the disability and figure out what is to be done, which is the final conclusion. So finally, if I'm studying DIA1, I am getting that these are the affected areas and which can be taken care with for what type of drugs or what type of therapeutic intervention should be given to autistic children. So these are some of the structure prediction tools and techniques, some of the genomics and protein tools and techniques, data visualization tools and techniques. And this is my project too, which I am supposed to do by July. This is when I'm giving my autistic children to eat quinoa, the highest source available for protein. So what is the development in their brain? And these I'm studying with the help of machine learning. So this is my project number 2. Okay, these are the references I have done. So point is, now finally what I am going to figure out, what is the results, is this valid? So this modeling is modeling part, but biology is a different thing, and for that the expertise of biological thinking has to be done. So this is what, if someone is interested to have the questions and collaboration, kindly do contact me. Thank you. I hope you all have enjoyed the session.
Thank you, Dr. Richa Mishra. Indeed, it was a very interesting approach that you've been taking, especially in a very need area, which is autism, essentially. Protein structure and prediction is very high interest research area. I think a couple of, I think it was a year back or so, Google's DeepMind or AlphaGo had their first shot at giving up protein structure, and that was a big news item essentially, and because that has implication into drug discovery and drug development. There's one question from Dr. Anju Arya in terms of how can we identify the sequence of proteins in different structure with the help of computers and only some step for computational access. Can you briefly talk about it?
Yeah, I have to go in like little hurry about it. So once you are going for machine learning part, the computation part, we need to understand the previous methods that will going to reveal us the profiling part. Using the multiple sequence alignments could accurately detect homology. So we need to understand the homology modeling first, and we can go with the pairwise sequence alignment generation method. The sequence alignment involves arranging the protein chain and the sequence amino acids in the matrix with the gaps inserted to allow similar or identical components to be allowed in a successive columns. So we need to understand this will help us to break the amino acid sequence up to a bitwise chunk for the model. And this will gives us the facility to identify the similar or the identical section, which can be result of the structural or evolution relationship with what we are looking for in the sequences. So when we are looking for a structural alignment in the form of sequence alignment, the attempts, there has to be the structuring of the polymer has to be understood. There has to be three-dimensional conformation. And for that, we are going to use a dynamic programming problem solving technique that breaks down the large problem into a smaller one. And the smaller problems are solved and in the optimal way to get the clear picture in the larger way. So then we need to formulate classification modeling and homology detection. Apart from that, there are some of the score calculation part. So these things has to be done as in the modeling, I hope. And at the end, we have to look for the k nearest neighboring algorithm for the clustering part. So the similarity level we can conclude with where the same type of clusters we are getting. So this is how we have to go step by step.
Great, thank you. So in the interest of time, I will have to restrict the questions. So what we will be doing, if you have any questions, we have a dedicated WhatsApp group for this FDP. You can pose the question and I'll route it to Dr. Mishra. If Dr. Mishra permits, we have a presentation, and that can be, if she permits, we can share that presentation within the group. So thank you once again, Dr. Mishra. It was really a refreshing take on how protein structuring is being linked to autism and what could be opening up to new avenues of drug discovery. Dr. Mishra, as she mentioned, she's very open to collaboration. So if a lot of my pharmacy faculty colleagues are out here, and this could be a very exciting area of collaboration where the expertise of yourself into pharmaceutical sciences could be matched with what's going on into the machine learning and artificial intelligence. So I think it will be a good area for collaboration. I like to motivate all my faculty colleagues from in pharmaceutical sciences to kind of critically look into that. It's a good area.
Yeah, actually, it's very upcoming one also. Like in India, yes, there is a good and large scope to go with. Yes. As there is actually a need that collaborated projects should go on, because though there is a limitation, I'm not from the bio side or the drug side, though I know the machine learning side, so it can be clubbed well and the projects can be taken care of.
Absolutely. I mean, I was looking at the Stanford's, art site for healthcare and artificial intelligence in healthcare. Half of the positions are, you have physicians, you have scientists from biomedical sciences, and half of them are core engineers. So obviously that field has to grow and will grow only with a joint collaboration and interdisciplinary research work. So with that, thank you so much once again, Dr. Mishra. Over to Professor Abu Sufyan for the closing announcements, if there are any.
Yes, sir. Thank you, Dr. Mishra, and all the dignitaries for making the day one of 1 week refresher program more fruitful. We are done with today's session. Feedback and attendance form we have already released in the chat box. It shall be remain active for 20 minutes. Also, those who have not yet joined WhatsApp group are requested to please join this group for further smooth communication. Tomorrow we have very exciting session by Dr. Ahmed Elinger, the Faculty of Computer and Artificial Intelligence, Beni Suef University, Egypt. It will start sharp at 7 p.m. See you tomorrow. Stay safe and stay blessed.
Thank you. Thank you so much. Thank you, everybody.
Thank you.